So this is the segment 3 of the chapter 3 outline. First, we're going to start off with the fact that the cell membrane is made up of fatty molecules that are used to repel water. Ions must pass through the membrane-spanning ion channels because they are surrounded by water molecules. And the potassium ion channel has an inner surface that contains oxygen atoms, and this allows potassium ions to fit but not sodium ions. Channelopathies is a medical condition that arises from abnormalities in the form and function of ion channels as a result of mutation of the genes that encode those channels. Sodium channelopathies result in seizures, muscle disease, and cardiac ailments. Synapses cause graded local changes in postsynaptic membrane potential. First, the action potential is spread down the axon. Then the axon splits into different branches in which the action potential is recreated in each axon branch until it reaches the terminal of each branch. Next, the electrical signal of the action potential is changed into a chemical signal as the axon terminal releases a neurotransmitter. Synaptic delay is the brief delay between the arrival of an action potential at the axon terminal and the creation of a postsynaptic potential. When a presynaptic neuron fires, it shows a normal action potential and causes depolarization in postsynaptic neuron. The inhibitory neuron fires. It shows both normal action potential and hyperpolarization in postsynaptic neurons. Postsynaptic potentials. A local potential that is initiated by stimulation at a synapse can vary in amplitude and spreads passively across the cell membrane, decreasing in strength with time and distance. When integrated, this massive array of local potentials determines whether the neuron will reach threshold. Therefore, generate an action potential in the zone. Excitatory postsynaptic potential, a depolarizing potential in the postsynaptic neuron that is caused by excitatory connections. This increases the probability that postsynaptic neurons will fire an action potential. The combined effect of many excitatory synapses is needed for action potential. All neurons use the same type of propagated signal. Meanwhile, an inhibitory postsynaptic potential, IPSP, is a hyperpolarizing potential in the postsynaptic neuron that is caused by inhibitory connections. This decreases the probability that postsynaptic neuron will fire an action potential. When a hyperpolarization moves the cell membrane potential away from the threshold, this is caused by inhibitory inputs fire. Postsynaptic effect is an increase of the resting membrane potential, and it results from the opening channels that permit fluoride ions to enter. Fluoride ions are more concentrated outside the cell, and then they rush in and make the cell negatively charged. Excitatory and inhibitory presynaptic neurons work in the same way with one exception. They have opposite effects on the postsynaptic cell. One factor is that some transmitters generate an EPSP in the postsynaptic cell, and others generate IPSP. EPSPs and IPSPs spread passively over the neuron, decreasing in strength over time. The same neurotransmitter may be excitatory at one synapse but inhibitory at another, and it depends on the receptors. Complex behaviors require neurons to integrate and transform messages they receive. Information processing. Each neuron adds and subtracts the myriad inputs it receives. Since some potentials excite while others inhibit the hillock, the effects cancel each other out. Net effect is the difference between the two. A neuron will subtract the IPSPs from the EPSPs, and no action potentials will occur. This can also be influenced by distance. Usually the convergence of excitatory messages from a lot of presynaptic neurons is needed for a neuron to fire an action potential. Spatial summation is when the summation at the axon hillock of postsynaptic potentials from across the cell body. If the summation reaches threshold, an action potential is triggered. Temporal summation is the summation of postsynaptic potentials that reach the axon hillock at different times. The closer in time the potential axon occurs, the more complete the summation. It results in a neuron without input. If EPSPs arrive continuously, the sum and the postsynaptic cell eventually reaches threshold and produces action potentials. Too much time in between results in the fading out of the EPSPs before the next cell comes. You could have EPSPs from two different synapses pushed together to fire the target at the same time. The overall postsynaptic effect is graded in size. Dendrites in cell bodies can induce postsynaptic signals. They augment the receptive surface of the neuron, and it increases the amount of info the neuron can take in. In some types of neurons, synapses that are distant from the axon hillock compensate by making larger postsynaptic potentials. Some neurons have dendritic integration zones instead. The transfer of info across a synapse is called a synaptic transmission. This occurs in a series of steps. First, the action potential arrives at the axon terminal. Next, this depolarization opens voltage-gated calcium channels in the membrane of the axon terminal, and the calcium ion to enter the terminal. Then the calcium ion causes synaptic vesicles filled with neurotransmitters to fuse with the presynaptic membrane and rupture, releasing the transmitter molecules into the synaptic cleft. Transmitter molecules then cross the cleft to bind to special receptor molecules in the postsynaptic membrane, leading to the opening of the ion channels in the postsynaptic membrane. Ion flow creates either a local EPSP or IPSP in postsynaptic neurons. Then after that, the synaptic transmitter is either inactivated by enzymes or revived by transporters. Finally, the synaptic transporter may also activate presynaptic autoreceptors. There are several specialized proteins that mediate exocytosis. The acronym SNARES includes two types of snares, V snares and T snares. Next we'll get into ligands. And at synapses with ligands occur at synapses with acetylcholine as a transmitter. It fits into ligand binding sites in receptor molecules located in postsynaptic membranes. The nature of postsynaptic receptors at synapses determines action of transmitters. Endogenous ligands include neurotransmitters and hormones. A curare is an arrowhead poison that blocks ACH receptors on muscles, which causes paralysis. Bungaratoxin is also an ACH receptor blocker. Agonists are poisons that act like a transmitter at a receptor. This includes nicotine and muscarine. Antagonists are molecules that interfere with or prevent the action of a transmitter.